Hello everyone, I'm Ruba Bayram, a first-year medical student and a member of PACademia. Today I'll be discussing the chapter Hypersensitivity and Transplantation. I'll be dividing the video into two parts. Today, this video will be about the hypersensitivity. The next video will be about transplantation, inshallah. So let's dive into this right away. So hypersensitivity, the definition is excessive or inappropriate immune response to any antigen, and it can cause host tissue damage. But the main point is that it's excessive and inappropriate. Now there's four different types. The first three types of hypersensitivity is going to be antibody-based, so it's going to be with the use of antibody, whereas the last one, type 4, is going to be cell-mediated, so it's using T cells. It's not using any antibodies, okay? So the first three, antibodies, the last one, no antibodies. Let's look at them in a broad idea. Now, when I talk about type 1 hypersensitivity, the main three things that I want you to focus on, and it's very important. Firstly, is that it's immediate, it's happening really quickly within several minutes to hours, that it is allergic, it's relating to the allergy. And then the third one is that it's going to be about the IG. So we're using IG. So three things. When you see in the question, immediate, allergy, IgE, you automatically know that it's going to be type 1. In type 2 hypersensitivity, now let's make it clear. The first three is going to be what? Antibody-based, right? Now type 2, is it going to be antibody-based? Yes, it is. In this case, I'm going to be having the antibody, and it's going to be binding to the cell membrane or extracellular matrix. Okay, so I have antibody binding to the cell membrane or extracellular matrix. So the antibody in this case, will it be IgE? No. In this case, the antibody will be IgG or IgM. That's the difference here. The type 1 hypersensitivity, they use IgE. It's immediate and it is allergy. Type 2 hypersensitivity, I'm using IgG, IgM, or IgM, okay? And it is going to be the antibodies will be binding to the cell membrane or the extracellular matrix. Now, in type 3 hypersensitivity, am I using antibody? Yes, I am using antibody. The antibody is going to be what? Again, it's IgG or IgM. But in this case, the antibody will be binding to soluble antigens. So antigens that are going to be found within the blood vessels, right? They're going to be binding to them, and they're going to be forming the complexes. But in this case, the antibody will be binding to soluble antigen. That's the difference. In type 4 hypersensitivity, is it going to be antibody-based? No, right? I mentioned the last one, who type 4 hypersensitivity will not have any antibody. In this case, we're using T cells or immune cells, and they're going to be directly attacking the host cells in the absence of my antibodies. So again, we have four types. The first three are going to be antibody-based. The last one is not. The first one, type 1, is going to be three things I want you to remember: immediate, allergy, and IgE. The second one is going to be antibody-based, IgG or IgM, and it's going to be binding to the cell membrane or extracellular matrix. The third one, is it antibody-based? Yes, it is. It's going to be binding to what? Soluble antigens. Last one is going to be T cells. We're not using antibodies. So again, type 1, allergy. We can see that there's IgE, and it's going to be really fast. Type 2, we mentioned, is going to be also antibody, that is IgG or IgM, and is binding to the cell membrane or extracellular, as we can see, cell surface antigen, right? Type 3, is it antibody? Yes, it is. What is it? It's soluble antigen. And we mentioned it's in the blood vessels, as we can see here, it's showing that it's a soluble antigen. Type 4, is it going to be antibody-based? No, it's not. It's going to be what? It's going to be T cell based, so cell mediated, right? Now, let's talk about type 1 hypersensitivity more in detail. So type 1 hypersensitivity, as I mentioned, there are three things that I want: allergic or allergy, immediate, and IgE, correct? Now, here immediate, what does it mean? As you mentioned, it's minutes to hours, so it occurs really fast. Like in the case of asthma. Asthma is a type of type 1 hypersensitivity. It is it's an allergic reaction to something, to an allergen, right? Maybe to a pollen, like to pollen, right? We can have these reactions, and it's very fast. We see patients who have an asthma reaction, for example, it occurs really quickly. They'll start to become very difficulty breathing, itchiness, right? As you mentioned, IgE antibodies. Now in this case, type 1 hypersensitivity is actually divided into two different steps. There's the sensitization and there's the challenge. In the sensitization, what I'm trying to say is that this is the first time I get exposed to this thing. Sensitization is the first time I get exposed to this allergen, and no reaction will actually happen. I'm just getting exposed to it. And the second time, which is the challenge, no, next time I'm being exposed to those re-exposure, this is where the reaction will happen. I will be discussing them now more detail so we understand. And sensitization, as I mentioned, is the first exposure to this allergen. Again, you can see allergen, it's mentioned again, so you always remember it's allergy, never forget. Okay, this is the first time I get exposed to this. What happens? I'll start to produce what? IgE. Remember we mentioned that the type 1 hypersensitivity is IgE. So now I'll be producing IgE. IgE will then start to bind to Fc receptors that are found on the mast cell. This step is the sensitization step. What did we mention sensitization? The first time I get exposed to the allergen, am I having any reaction? Will I see anything? No, no reaction. So sensitization, first time I get exposed, my body will start to produce IgE, then the IgE will start to bind on Fc receptors that are found on the mast cells, and they form this complex. This is the complex. The complex of the mast cells with the Fc receptors right here, and then with the IgE antibody. This is the complex. This is sensitization. Challenge is what? Challenge is when I'm going to be having a repeated exposure to this allergen. So it's the second time I'm going to be exposed to this, right? Second time I get exposed, as we can see, now I have already the complex. I already have the complex here. This is the complex. So now this is the time I'll actually have the reaction because I have the complex. The first time I didn't have the complex, so no reaction will happen. Do you understand? And as we can see, this is the allergen right here. It will bind to it now, and this will trigger the reaction. In this case, is the mast cells will start to release the contents it has, which in this case is histamine and also the cytokines. We know the histamines are going to be what? They're vasodilators, right? Histamines are vasodilators, and that's why we see that the patient is going to be red in color, their itchiness, right? Now, one thing we want to mention that in the IgE, we mentioned in the sensitization, IgE is being produced, but how is it being produced? The IgE is being produced through two different ways. The T helper 2 cells, okay, and the interfollicular secreting helper T cells. So two different ways. But the same thing that I explained is here. So these are the ones that are secreting the IgE. Once the IgE is being formed, it will bind to the Fc receptors that are found on the mast cells. This is sensitization, correct? Because here, the first time I get exposed, my body will be producing IgE through what? Through the T helper 2 cells and then the follicular helper T cells. And then after this, the IgE that is being produced will bind to the Fc receptors that are going to be found on the mast cells, and they form this complex. Now, in the challenge, this is where I say this is the subsequent exposure. The second time I get exposed to this antigen, and I'm going to be having what? I'm going to be having the cross-linking of the bound, right? It's the bound. It's bound to the mast cell. And then, yeah, this leads to the activation and release of various mediators. Now, the T helper 2 cells, they'll be releasing interleukin 4, the 13, and even 5. So interleukin 4 and 13 will have the allergic reactions that we know, excess mucus secretions, intestinal motility. But the interleukin 5, we will see that they're going to be having or promoting eosinophilic inflammation. We start to see some eosinophils as well in here. Okay. Now, which we know eosinophils are used for allergies or parasites. We have studied this before. Okay, so it makes sense why they are also going to be involved. Now, type 1 hypersensitivity can be localized or systemic. Localized means that this is when the allergen is going to be entering. Where is it entering? Example is the asthma that we have just discussed. The asthma, in the case of asthma, the allergen is going to be entering through inhalation or through the respiratory tract. Like let's say in the pollen, anything it could be, right? But the idea that it can be in the respiratory passage. Could be intestinal wall, like in the food allergies, basically in the stomach, anything. But the idea is that it's localized. It's where this allergen is entering. This is where the reaction is going to be happening. As mentioned, the example is the asthma. Okay. We can also have it systemic. In the systemic, now this is where the allergen is going to be everywhere, and it's going to be in the blood vessels, right? In this case, like in the venom or in the toxins, right? This is the systemic. I'm having injected allergens. And so what I will have is that I'll start to see that there's anaphylaxis, okay? This is anaphylactic shock. In this case, that's very unique, is that I'll say that there's vasoconstriction and vasodilation at the same time. The vasoconstriction is going to be happening, now you'll be wondering, okay, vasoconstriction and vasodilation happening at the same time, I guess that doesn't make any sense. The vasoconstriction is happening in the arteries, whereas the vasodilation is happening in the veins. I just learned it as AC. You know the AC we use, arteries contract or constrict, whereas the VD, the veins will dilate. And now upon dilation, we will see that what happens is that the gaps in between the endothelial cells will start to increase. When this increases, as we know, what happens, the proteins and the cells are able to leave now. And because of this, now the fluid will also, or the water will also follow through because of osmosis. And this is going to be causing edema and swelling. This is what we see in anaphylactic shock. We'll see that the patient will actually be, they'll face edema even in the face swelling. We'll see it mostly. There's severe loss of this fluid and it's going to be accumulating and it's going to be very difficult for them to see. Actually, this is how the patient anaphylactic shock will look like. You can see there's so much this fluid that they can't even open their eyes, right? So this is the anaphylactic shock criteria. So type 1 hypersensitivity, allergies, IgE, and it's immediate. And we mentioned the examples. It could be the asthma, which is localized. Could be also food allergies is also localized because it's in the stomach. Or it could be anaphylaxis, it's going to be systemic. Okay, we mentioned the ways, there's sensitization, and then the what's the challenge? Okay, now type 2 hypersensitivity. Remember, type 2 hypersensitivity, is it antibody based? Yes, it is. But here the antibody is being used, what is IgM or IgG as we mentioned. It's not Ig. So we mentioned it is going to be using antibodies, yes. What antibodies am I using? IgM or IgG? Okay. What is it binding to? It's binding to the cell membrane and the extracellular matrix, as we mentioned. That's how it is. Now, in the case of these antibodies, okay, they bound to the cell membrane and the extracellular matrix. What happens after that? Okay, antibodies are bound. What do? Antibodies, as we know, they will do many things. First of all, they can activate the complement system. We know the complement system can be activated through many different ways. It could be through the classical pathway, the alternative pathway, the lectin pathway. The classical pathway is through the antibodies. So when there's antibodies that are bound to the cell membrane or extracellular matrix, the complement is going to be activated. And when the complement is activated, it can do many things. First one, it can do the inflammation, right? It can do the lysis through the MAC. It can cause the opsonization. So now the antibody is activating the complement for all these three things. On the opsonization, I'm going to be having phagocytosis with the macrophages and the neutrophils, right? And another thing the antigens will do is they're going to be activating the natural killer cells by the ADCC, antibody-dependent cell-mediated cytotoxicity. Okay, so the antibody, as you can see, it's not necessarily doing the action, but it's activating many different pathways to do the action of getting rid of the allergen or whatever through the neutrophils or the macrophages, through the natural killer cells. Okay, the complement is also activating more and more. An example of that is the blood group antibodies. Now, in this case, let's say I'm going to be having type A. My blood is type A. And I need blood transfusion. I lost a lot of blood. I need to some blood to be donated for me. But let's say by mistake, someone with type B antibody, type B blood, they gave me their blood. That's bad, right? We always know that we need to have blood transfusion within the same type, right, of the blood. The reason is because, as we know, we have taken this before, the type A is going to be having antigens of type A. It has antigens that say, okay, I'm type A, I'm type A, I'm type A, the antigens that are on the RBCs. But also, what I have is I have in my plasma cells, in my serum, plasma, I will have anti-B. So it's basically antibodies against the B antigens that are found on the patient with the type B. This patient type B will be having what? RBCs, and it will be having the antigens that are B, that says, okay, I'm B, I'm B, I'm type B, I'm type B, right? When I have anti-B, which are the antibodies against these antigens, when they bind together, when someone is going to be transfusing the type B antibody, I have already antibodies towards them. So what happens is that I'll end up binding to the transfused RBC, to the donated blood. My antibodies will be binding to it, and it causes it to be destroyed. It causes it to be hemolytic. How? Through, as we mentioned, the same way, the macrophages, the neutrophils, the natural killer cells, whatever way. But this is the method that we're talking. And that's why it causes hemolytic anemia. From the name hemolytic, lytic is going to be the destruction, right? Hemo, the blood cells are being destroyed. As causes anemia. Okay, so this is one example that we're talking about. Okay. And here this is the same thing I mentioned. We're having IgM or IgG that are binding to the cell surface or the cell membrane extracellular matrix. It's going to be activating the natural killer cells through the ADCC, or the complement, which is going to be causing the opsonization through the phagocytosis, or it's going to be the inflammation. So it's going to be recruiting the neutrophils and the monocytes, right? So these are the many different things. Now, another example, the first example we talked about the blood transfusion, which is the hemolytic anemia. The other example is that sometimes antibodies will be able to bind to receptors. So antibodies will bind to receptors. Now, this could be stimulatory or inhibitory. An example is that antibodies will be binding to the thyroid cells. Thyroid cells, they will be having receptors for thyroid-stimulating hormones. Thyroid-stimulating hormones from the hypothalamus. I don't want to get into details, but hypothalamus will be releasing thyroid-stimulating hormones. From the name, it are their hormones that will stimulate the thyroid, and then the thyroid will be releasing their own hormones. In this case, what happens is that the antibody will bind to this receptor. Here, this is my antibody. And this antibody will bind to the receptor. And what happens is that the antibody will act as if they are these stimulatory hormones, and they will continuously bind, because they're bound to it, they're bound to the receptor, they're continuously activating the thyroid. Continuously activating the thyroid. And so the thyroid will continuously be forming the hormones. And this is known as the Graves' disease, where the patient is going to be having hyperthyroidism, because the antibodies that are bound to these receptors will continuously activate them and stimulate them. So this is an example of stimulation. An example of inhibition is when I have the muscle cells, it has the acetylcholine receptors, right? And acetylcholine needs to bind onto the receptors. But in this case, what happens? Again, the same way, antibodies will bind to the receptors. But in this case, it's not going to be stimulatory. No, it's going to be inhibitory. In this case, it will prevent the actual acetylcholine from binding to the receptor. And so now the receptor will no longer be working. The muscle will no longer be activated. So I'm going to be having inhibition. The muscle will be inhibited. The muscle won't be working at all. So this is where I have myasthenia gravis. They're going to be like flaccid. They're paralyzed, okay? They're not working. They're not stimulated at all. So the type 2 hypersensitivity, I'm going to be having what? Antibodies. The antibodies are what? IgG or IgM. They're binding to what? Cell membrane or extracellular matrix. When they're binding, what do? They activate many different things. They activate the complement by the classical pathway. It's going to be causing the opsonization, which is then in turn causing the opsonization and the macrophages and the phagocytosis. I can have the inflammation, recruitment of other inflammatory cells. I can have the natural killer cells by the ADCC, right? Another way is that these antibodies will be binding to the receptors. This can be either stimulatory or inhibitory, and we gave the examples. The examples are also important, and these are mentioned, right? The Graves' disease, we mentioned the hyperthyroidism, the continuous stimulation, or the myasthenia gravis, which is the paralysis, as we mentioned, or muscle weakness, right? Because the muscle is no longer accepting any acetylcholine because the antibody is blocking it. It's blocking the receptor from accepting any acetylcholine. So now it's not working at all. It's paralyzed. It can't contract. Okay, in type 3 hypersensitivity, what we mentioned, let's go in بالترتيب in هذا. Type 3 hypersensitivity, is it antibody based? Yes, it is. What antibody am I using? So it is antibody based. What antibody am I using? IgG or IgM? Okay, it is against what type of antigen? The soluble antigen. Soluble antigens. Okay? This is exactly what we're saying, soluble antigens. Okay, now the antigen, it could be exogenous or endogenous. Yeah, this antigen, the soluble antigen, I could have gotten it from outside sources or from inside. The outside sources is basically through the vaccine. We know the vaccine, it's exogenous. Basically, I have a small, inactive, weak antigen is going to be placed and introduced in the body. This is the same. So I can have localized reactions again, or systemic. Localized reaction is known as Alpha's reaction. Here, I'm going to be injecting this antigen under the skin. It's intradermal injection, and just a small amount. And because it's localized, I'm actually going to be seeing it in 4 to 6 hours. Yeah. It's going to be faster. What am I saying in localized? I'm just going to be seeing on the skin, it's going to be painful. It's going to be slightly raised, right? So this is what's mentioned. Painful, it is localized, it is edematous, right? And it's a lesion, so slightly raised, right? In this case, as we can see, this is in the blood vessels, so already was the soluble antigen. The antibody is binding to it, and because of this, it forms this complex and it starts to deposit. Now, again, the same way, I have antibody binding to the antigen. What is it going to be stimulating? It's stimulating the activation of what? The complement, the recruitment of the other inflammatory cells. So now I will see that the neutrophils will come and whatnot, right? And this causes the inflammation of my blood vessels, vasculitis, okay? Now, it could be systemic. The other example is the serum sickness, right? Systemic is now more within the whole body system. I'm going to be having these examples. But the same way here, the exogenous antigens is going to be what? When I'm going to be having a vaccination. The vaccine, as we know, usually is going to be from like outside sources, like from horses, let's say, or whatnot. Now, this sometimes can be, now you might be wondering, okay, in the vaccine, I'm supposed to be having a reaction. What's wrong with that? The vaccine, I know it's an antigen. I'm supposed to be in my body, and I'm supposed to be having a reaction to it. Why is it called the hypersensitivity? No, this is different. The vaccination, I'm having a reaction towards it. This is good. But when I'm having remember, hypersensitivity is what? Excessive or inappropriate immune response. So I'm having an excessive response to this antigen. I'm not supposed to be reacting this severely to this vaccine, right? So this is the type of exogenous antigens. Exogenous is from outside. The example is the vaccine. Example, we mentioned the Arthur's reaction, which is localized, is just an injection locally. Or systemic reactions is going to be in the case like of the vaccines we mentioned, it's going to be causing the serum sickness. The same way we mentioned, now the antibody will be binding to the soluble antigen and is going to be stimulating what? It's activating the complement, and what activates the complement, it causes the vascular injury, which we mentioned is the inflammation, the vasculitis, right? Okay, it will attract the inflammatory cells, as we mentioned. And here we mentioned it's the consequence of treatment with animal-derived anti-sera, so basically antibodies, anti-sera and serum, that's why I mentioned. Antigens, and we mentioned examples, the horses. I mentioned the soluble antigen could be exogenous or endogenous. Exogenous, we mentioned it. The endogenous is going to be what? Self-antigens. So antigens, if they're endogenously from me, self-antigens. When we think of the self-antigens and immune response or whatever, is what? Is autoimmune disease. An example is the SLE. The SLE, okay, we're producing antibodies against my own antigens. The same way, the antibodies will bind to the soluble antigen. They start to deposit, they will start to form the vascular inflammation or vasculitis. It will activate the complement, right? The main important things is the deposition of these complexes that are being formed, that there's going to be vasculitis or the inflammation of the blood vessels. There's a soluble antigen, and the examples are very important as well, right? Now, these immune complexes, sometimes they form in the immune complexes, and we mentioned they're going to be deposited. Sometimes it will be deposited in the blood vessels near the kidney or whatever. It can cause nephritis, the inflammation of the kidney or kidney failure, right? These are all examples as well. Okay? Now, type 4 hypersensitivity, again, is it involved with antibodies? No. So, no antibodies. So, then what is it? It is going to be T cells. So, that's why we mentioned T cell mediated and does not involve any antibody. Now, again, we have localized or systemic. A localized contact dermatitis. This is an example from the name, contact dermatitis. Contact and dermatitis, my skin. So when I'm going to be coming in contact with certain things like metals or chemicals or plant poisons or whatever it is, and as we know in my dermis, I'm going to be having CD8 cells, right? I'm going to be having T cells in my epidermis, right? Or in my skin. So again, it's going to be just that reaction where my T cells are being involved. An example is the contact dermatitis. As we can see here, it takes 7 or more days. Whereas, for example, the others, it usually just like, for example, type 1 hypersensitivity, several minutes. The others as well won't be taking much time. The reason why is because this is T cell mediated. It's T cell. I'm using T cell. There's no antibody. And T cells need first to be activated, then they need to be proliferating and then differentiating. So it takes some time for them to do this, right? Now, another type is delayed hypersensitivity. As we can see now, in this case, it's non-topical. That one, the contact dermatitis, it was topical, right? It was locally on the surface of the skin. The delayed type hypersensitivity, no, it's non-topical. And now I'm going to be having the intracellular organisms. We know most of the times these are going to be involved with organisms like bacteria, the fungi, these things, right? Again, it might take some time as well. The example is also TB skin. TB skin test is basically, they want to test if you're going to be having tuberculosis, basically. So they're going to be injecting it. And if there's going to be a certain reaction towards it, right? If there's going to be a reaction towards this mycobacterium, which is going to be causing the tuberculosis, I'm injecting the bacteria right now. intracellular organism. And that's why we know it's okay, it's T cell, because I'm injecting intracellular organisms here, and we're going to be using T cells. Okay, so the main thing is know the examples, like the TB skin, the delayed, the contact dermatitis, and what is T cell mediated. As we can see, T cell mediated. Now it could be through the CD4 or CD8, but mostly it's through the CD8. CD4, as we know, CD4 cells, they are going to be releasing cytokines to cause inflammation. And then, whereas the CD8 cells, no, they cause the direct killing of the by apoptosis, right? Another example of the hypersensitivity is the graft rejection, which we're going to be talking about it really soon in the next video. But as you can see, these are all the examples as well. So this is the summary. This summarizes the main things that we mentioned. So let's go over them. T helper 2 cells, what do? They were the ones that released the IgE. IgE, after they're being released, what do? They bind to FC receptors, right? These are the FC receptors that are found in the mast cells. And we mentioned that actually T helper 2 cells release what, if we remember, the interleukin 5, which causes eosinophilic inflammation. We mentioned generally eosinophils are going to be involved in the allergens, right? And we mentioned type 1 hypersensitivity is allergy, it's immediate, right? Okay. Type 2 hypersensitivity, we mentioned, is going to be use of IgM or IgG, and it's going to be against the cell surface or the cell membrane and the extracellular matrix, correct? And we mentioned because of this antibody, it will activate the complement, it will activate the opsonization, right, and phagocytosis, and the recruitment of the other leukocytes. Okay. Another example we mentioned was what the abnormalities where it binds to the receptors, correct? Do you remember? The hormone example was the thyroid hormone, it's stimulatory, causes the Graves' disease, the hyperthyroidism, or the neurotransmitter receptor signaling, the one about the acetylcholine, the myasthenia gravis, which is going to be what? The case of the muscles, where they're not going to be stimulated at all. Type 3 hypersensitivity, what is it going to be? Now, the type 3 hypersensitivity is going to be immune complex mediated, because we're forming this complex where it is going to be a soluble antibody, a soluble antigen, and it's going to be forming these complexes, which are then going to be deposited. So here again, we mentioned it's IgM or IgG, and we mentioned it's going to be deposition into the blood vessel. Okay, and again, it's going to be activating the complement. Now, in type 4, lastly, we mentioned is going to be the T cell. It's T cell, it's not antibody. Okay, remember that. We mentioned it could be CD4 or CD8. CD4 is the cytokine, it releases the cytokine. CD8 is going to be, in the case of the cytolysis, is going to be releasing the spirophorons and whatnot, and going to be actually causing the apoptosis of the cell. Okay, so this is it for this lecture. I'll be shortly making another one about the transplantation, so watch that too. And that's it. I hope you guys understood. If you have any questions, please write down in the comments. Yes, thank you.